All right, so my name is Christopher Barnes. I'm a new assistant professor in the biology department at Stanford. And so my lab is basically interested in viral host pathogen interactions. And so this is a report issued in September 2019 by the Global Preparedness Board showing us that we are at risk of devastating, zoonotic transmission that can lead to pandemics, right? So there's many viruses, many bacteria on this list that we are at risk of, spilling over into our population, leading to future pandemics. So how are we preparing for this? So my lab uses basic science as well as translational, different types of techniques to understand viral host interactions to advance human health. And so in essence, we work with clinicians, we work with doctors around the world to establish patient cohorts, whether these be people that are chronically infected, like those in the case of HIV, or people convalescent for viral infection, like those from SARS-CoV-2. Our lab is interested in adaptive immune response, and so we're really focused on really the B cell response after infection. Begin to characterize these responses to understand what are the correlates of protection that can lead to better therapeutics or vaccines. Our lab specializes in structural biology techniques, so using things like cryo-EM, crystallography, and moving more towards cryo-electron tomography to begin to understand how these viruses are infecting cells, forming replication complexes and organelles to help us understand exactly what's going on in that cellular context. We then take this structural information and use directed evolution techniques, mammalian display, surface display, to either engineer antibodies to be more potent or more cross-reactive to help cover a larger breadth of viruses or within the same family. Or we use this information to help design immunogens. And these are things like that we can present on a vaccine platform that we can then deliver into animals to elicit a robust immune response to what we want. Then, of course, we move all this back into animal models. And so we work, again, in our lab as well with collaborators to begin to understand exactly how our designed immunogens, whether it be vaccine immunogens, displayed on nanoparticles, are being biodistributed across the animal and stimulating germinal center responses, or we think about how we can use antibodies and challenge animals to see how they are being protected. And of course, within all of this, there's a lot of feedback happening. So we go back and forth to help further characterize our immunogens using structural biology or understanding the responses that are coming out. And so right now in our lab, we focus on HIV. So as a postdoc, I was really in the HIV field, helped characterize a new epitope called the silent face, which has antibodies directed towards it that are more amenable, we think, towards vaccine design in the HIV space. We're also working in the coronavirus space still, and so recently we have a paper coming out where we identify antibodies that bind to the fusion peptides as well as the stem helix, which actually have breadth of binding across the whole family and genre of coronavirus. So we now think that we can actually make pan-coronavirus therapies or vaccines. We're also interested in moving into the flavivirus space. So obviously, most of the world is endemic for one flavivirus, and neurotropic flaviviruses are on the rise, especially here in the US, like in the case of Powassan virus, which is tick-borne. So we're interested in that, as well as understanding autoantibodies and disease manifestation. So thank you so much, and looking forward for collaborations.